Good morning and happy Friday. Just a few reminders here. Participation 1 was due this past Wednesday at 11:59 p.m. If you forgot to complete that, please just send me an email and I'd be happy to reopen that for you. So this next week, we will begin the flipped classroom model. And so before class, please watch the videos. These are listed in the schedule of readings and videos. The team assignment form is due this Friday, so please complete that and submit that so that we can make the teams this weekend. And finally, homework 1 is due this coming Wednesday, design process and population dynamics, and participation question 2 is due at the same time. So this morning what I'd like to do is talk about population dynamics. The material for this unit comes from section 5-5 from Principles of Environmental Engineering and Science, a textbook that I co-authored with Dr. Mackenzie Davis, retired professor from Michigan State University. And we'll look at bacterial population growth, animal population dynamics, and human population dynamics. And this reading is in the folder called Assigned Readings. I changed the title. And it's on non-kappa. So as I mentioned, what we want to look at is population dynamics. So this is a study of the changes in the number and composition of individuals of a particular population within a study area and the factors that affect these numbers. So this can include any organism, an organ from small as a bacteria to as large as a whale in the ocean. And we can define the study unit based on whatever question essentially we're addressing, whatever issue we're addressing. So that study unit can be biological, it could be a lake, it could be a river, it could be the open ocean, it could be geographical, so it could, or political, so it could be a political boundary of a community, or it could be an engineered system. So it could anything from an island, which would be geographical, a city, so political, or an engineered system. So in this course, the systems that we will most commonly look at are, let's kind of combine it as geopolitical. We'll talk about service areas for a water treatment plant or a wastewater treatment plant. And we'll talk about bacterial populations, and this can be pertaining to reactor design and operation. It can be pertaining to a water distribution system. Could be the organisms in a biofilm in a distribution system, so in the inside of a piping network, and predominantly that's made up of bacteria, but it can also include, for instance, amoeba. And we could talk about bacteria in pertaining to site remediation, where we're relying on bacterial decay.
So if we're looking at bacteria growth requirements, we need to think about what do these organisms need? What do organisms need for growth? Let's try and write this in the chat. What are some of the I'm going to call it factors. So food, okay? So food, space, in some cases sunlight, water. So we can divide it. You've mentioned a number of factors. All organisms need a terminal electron acceptor. What is the terminal electron acceptor required by humans? In terms of the energy cycle, what is the terminal electron acceptor required by humans? Exactly, oxygen. Okay. Other organisms can use nitrate, sulfate, or methane. And in terms of energy obtained, the energy that's obtained from respiration, it decreases as we go from oxygen down to methane. All organisms need some sort of macronutrients. The macronutrients are carbon, nitrogen, and phosphorus. So we often talk about a carbon-nitrogen phosphorus ratio because those are the macronutrients. They also need micronutrients. These include trace metals, vitamins, and they need a proper environment. So they need, as mentioned, water. They need an optimal temperature, and they need an optimal pH. And I'll just add this, they need an energy source. So for some organisms, those that are phototrophic, they need sunlight. Those that are heterotrophic, like us, we get our energy from carbon. And there are some organisms that can get their energy from CO2, so they can get it from inorganic carbon, others from organic carbon. So we are heterotrophic, we get our energy from organic carbon. When we talk about temperature, we can categorize organisms based on temperature. And the two types of organisms most typically most important to us as environmental engineers are the mesophils, and these are optimal at about 20 to 40 degrees, and the thermophils, which are optimal above 40 degrees C. There are also psychrophils, and these typically are optimal from about 20 degrees C to 10 degrees C. And then there are the hyperthermophils, which are optimal or have an optimal growth above about 60 degrees C. So this gives you a range. So for instance, mesophils and maybe some of the psychrophils are the ones that are most optimal when we talk about wastewater treatment in a wastewater treatment plant in an activated sludge basin. The mesophils, kind of ranging to the thermophils, are the ones that are most optimal in a anaerobic digester, which is heated, because we want a temperature there typically around 35 degrees C. So it's kind of the high end of the mesophils, the low end of the thermophils. We can also talk about organisms in terms of their respiration. And here we talk about aerobes. So those are organisms that require oxygen. We are an obligate aerobe. There are bacteria that are obligate aerobes. They can only grow in the presence of oxygen. And then anaerobe can only grow in the absence of oxygen. And an obligate anaerobe cannot survive in the presence of oxygen. And then we have organisms that are referred to as facultative. And these are much more able to adapt to different environmental conditions. So they can switch their respiration, whether they're in the presence of oxygen or the absence of oxygen. Typically, they can't grow in absolute anaerobic environments. They need a little bit of oxygen, but they can switch respiration. Any questions? When we're looking at bacterial growth in a pure system, and this is a closed system, so think of a beaker. If you actually, you made soup and you left the soup out, over time, bacteria will grow. Okay. Initially, there's low levels. As the bacteria become acclimated to the environment, the growth accelerates. It then reaches a log phase. Growth slows down. We have no net growth. And then we have a death phase. So we have a declining population. So the growth rate is negative. It's declining. Okay. The stationary phase, the growth rate is equal to the death rate. So they're dying off at the same rate that they're growing or proliferating. And then we have a log growth. The point at which this decline occurs is referred to as the capacity, the carrying capacity. So this point here, we can talk about this as the carrying capacity. The system can no longer support growth. Why does the system move from growth, log growth, or exponential growth to a stationary phase? What's happening at that point in time? Okay, good. Nutrients are being used up. Excreted toxins are building up. There's limited resources. So our nutrients become limiting. Okay. Toxic byproducts build up. What else? Is there anything else that happens? When organisms become stressed, what typically happens? What part of the semester are you most likely to get sick? Typically. The end around exam times or around midterms. Why? Because you're stressed. When you're stressed, and this goes for the red pine, the cedar, or us, okay? When we're stressed, disease organisms are much more likely to proliferate. You get sick, okay? And that says that the fact that the nutrients are becoming limiting, the fact that the toxic byproducts are building up, results in stress, which makes the organism more susceptible to disease. In a community, as the population is growing, why might the population or the growth rate start to decline? So if you're looking at a city, township, some sort of a community, why might the population growth rates start to decline. Exactly. Limited space, congestion, space limitations. Okay. Population, we can go out for food. Okay, so it's not like nutrients necessarily become limiting, but space becomes limiting. When we're looking at bacteria, we model that based most commonly with a geometric progression. Bacteria divide by binary fission. So one organism becomes two organisms become four organisms become 8. So it makes sense from a biological perspective to use a geometric progression. And the equation is just that the population is equal to the initial population, which is typically at this point here after accelerated growth phase, times 2 times the n number of generations. So n equals the number of generations. Okay, so as I said, we start with 1. That one organism divides, we have 2. Those 2 organisms divide, we have 4, 8, 16, etc. Okay, this is true for closed system. We're only looking here at the log growth phase. Okay, so we're assuming unlimited space, unlimited nutrients, unlimited energy source. So we can look at this in terms of works in terms of yeast. Ever since, actually for the last year and probably year and a half, I've had a sourdough starter. It's yeast. Okay, those yeast cells divide and they form 2 new cells about every 5 minutes. So if you place 10 to the 5 cells in a suitable environment, so I provide them water, I provide them food in the form of flour. Okay, I have a closed system, it's in a jar. So if I start with 10 to the 5 cells, how many cells do I have in 30 minutes? So how do I determine that? So P is equal to P0, 2, because they're dividing by binary fission, times n. And n is the number of generations. So n is 30 minutes, and there's 5 minutes per generation. So that's equal to 6 generations. So P is equal to 10 to the 5, 2 to the 6th, which is 6.4 times 10 to the 6th cells. Okay. And we could also determine that we start with 10 to the 5, divides, so we now have 2 times 10 to the 5, 5 minutes later, 4 times 10 to the 5, 10 minutes later, 8 times 10 to the 5, 15 minutes later, 16 times 10 to the 5, so now we're up to 20 minutes, 32 times 10 to the 5, 25 minutes later, and after 30 minutes, we have 6.65 times 10 to the 5, or 6.5 times 10 to the 6. So we can look at it either way, and get the exact same answer. If you don't, you've done something wrong. Now, the challenge is that pure cultures don't exist in nature. They're not going to exist in the wastewater treatment plant. They're not going to exist in the distribution system. They're not going to exist in the soil system. They don't even exist in the pot of soup that you made and left out because you forgot to put it in the refrigerator. And as we mentioned, log growth only occurs when the organism has unlimited nutrient, unlimited space. There's few external limitations. And we've talked about this in terms of what limits growth. So before, okay, we've said limits in terms of food, shelter, we've mentioned that. Okay. We've mentioned disease. In a mixed culture, we're going to have predators. We're going to have competition. And I spelled competition wrong. For food, space. We may have unfavorable physical conditions. For example, in a wastewater treatment plant, in an anaerobic digester, two different types of organisms. You have the acid forming and you have the methanogens. And the methanogens are very, very susceptible to pH. So if you've got too high a growth of the acid forming organisms, the pH, and not enough alkalinity, the pH drops and kills off the methanogens. So what's really killed off those methanogens isn't limitations in terms of food. It's not limitations in terms of space. It's actually an unfavorable physical condition. So the pH drops and the organism dies. If you have multiple organisms, which is the organism that is most likely to survive? Which is going to be the more successful organism? Okay. The one that can adapt to changing conditions. So adaptation is extremely important. Okay. So the ability to adapt. What else? Okay. Two organisms present. Start at the same concentration or the same number. Which one is going to proliferate faster? The one that has a shorter reproduction period that can metabolize the substrate more effectively. And better reproduction. So that's the more successful organism. And that's often a problem in site remediation, where you're relying on biological remediation. So one of the challenges of actually introducing an organism is that organism has to outcompete the natural organisms. Okay, we can also have exponential growth. Exponential growth assumes infinite resources. So we're describing this here. The change in the number of organisms with respect to time is equal to some rate constant times the number of organisms. We can integrate that from n0 to n and from time 0 to time t, and that is n sub t is equal to n sub 0 e to the rt, or n sub t over n sub 0 is equal to or the natural log n sub t over n sub 0 is equal to RT. Okay. If R is positive, we have growth. If R is negative, we have decay or decline in population. And this graph just illustrates this. So it's illustrated here with a doubling time of 1 day, and R with a R equal to 1, so that rate constant equal to 1.0. After 10 days, start at 1, day 0, we've got 1 organism. Okay. After 10 days, with an R equal to 1 and a doubling time of 1 day, we have 1,000 organisms. So N at 10 days is equal to 1,000. If R is equal to 0.9, then after 10 days, we have 455 organisms. If R is equal to 0.8, after 10 days, we have 230 organisms. And if R is equal to 1.1, after 10 days, we have 1,772 organisms. This R is similar to what we talk about as the R effective with COVID-19. It's slightly different, but it's the same sort of modeling. It's why we want, you can see very small changes in R can have a very significant change in the number or in this case, organisms. So we don't kind of think about that when you think about COVID. Again, like I said, the R effective is slightly different than this, but it's the same sort of concept. Small changes in R actually have a very significant impact on the reproduction rate and the number of cases we have in a community. We can also use a geometric or logistic model. This assumes, again, it's assuming infinite resources, but it's that growth occurs in discrete intervals. Shown here in terms of days. So we have a certain number of organisms on day 3. We've got an increase on 4. It's growing in discrete increments. So n sub t is equal to over n sub 0 is equal to lambda times t. T is the time step. So the growth is occurring in these time steps. Okay. If lambda is greater than 1, then you see an increase in population. If lambda is equal to 1, then there's no change and I apologize for these lines, and if lambda is less than 1, then there's a decrease in the population. Again, this assumes a closed system. So we don't have organisms that can move in and out of the system. Now, we can also have a logistic model with a density-dependent limitation. So essentially what we've done here is we're using the exponential model, but what we're saying is that there's we talked about a carrying capacity, okay? There's some limit to growth. And as the number of organisms approaches that carrying capacity, the rate decreases. So here, the change in the number of organisms is equal to the rate times the number of organisms times K divided by N over K. And K is the carrying capacity. And if we integrate this, then the number of organisms at any time t is equal to K times N sub 0 over N sub 0 plus K minus N sub 0 e to the minus RT. Okay. So what I've plotted here is this is K is equal to 10 to the 6, so 1,000,000. Basically, from the perspective we have here, there's unlimited growth, unlimited food, unlimited space. There's no limitations to growth, at least from the perspective we have here. Here, K is equal to 1,000. So notice the rate is increasing, but that rate decreases, the rate of growth is decreasing as we approach 1,000. And here, K is equal to 250. So we see it starts out the same for each of these systems, but here the maximum number is 250. So we're limiting growth. The Lotka-Volterra is a predator-prey model. So P is our predator, and N is the prey. And what you see here, okay, so we've got lynx and hares. So the hares are the prey, the lynx are the predator. So what happens? The hare population increases. The predator population is lagged behind. So notice there's a lag time. As the population of the hares increase, there's more food for the lynx, so their population increases. But eventually there's too many lynx. They're eating too much hare, too many of the hares, and the hare population decreases. And now, ultimately, the lynx population decreases, and it can just cycle through. So we can model this essentially with a logarithmic growth, but also a decay rate. So then here, the change in our predator population with respect to time is equal to some growth rate, and A prime is the attack rate, and C is the birth rate. times P sub n times N minus Q, where Q is the death rate, times P. And we can develop a similar expression for the prey, and that is equal to the growth rate of the prey minus the death rate times P times N. There's no attack rate for, essentially there's an attack rate right here, okay, times the attack, how quickly they're being attacked or consumed by the prey, by the predator. The exponential model is commonly used for human populations. And you can see this here. This is population of Pima, Arizona. It looks pretty exponential. So we can describe this as the population at any time is equal to P0 e to the minus RT. And with humans, that rate is equal to the birth rate minus the death rate plus the immigration rate minus the emigration rate. So B is the birth rate, D is the death rate, I is the immigration rate. So how at what rate of people moving into this community, and M is the emigration rate, just how quickly they're moving out of the community. One of the things to be careful of, though, is the time scale. If we look at the population from 1860 onward, it looks pretty exponential. If, on the other hand, we only look at the population from about 1960 and we plot it here, it looks linear. So the time scale plays an important role in this, in determining your rates. And this will become very important in the first part of your design project, because there what you will do is you have to determine the population. You can't design your system if you don't have an estimate or prediction of the population 25 years from now at the design period. What do you use? How do you determine that? And you can see that here. If we look at the population of Brighton between 1970 to 2000, it looks linear and it looks like it's increasing. If we look at it from 1970 to 2000, we might be able to use an exponential plot, or maybe it actually looks more like it is a plot with carrying capacity. Flint, we can do the same. If we look at the population growth in Flint between 1990 and 1960, it's increasing. If we look at it from about 1970 onward, it is significantly decreased. So that plays a very significant role. Okay, so this is population for Brighton. I was going to have you plot the data, but I've plotted the data for you. So here's the data plotted. And we've got a couple minutes. I think we've got time for this. I'm going to ask you the question. So me unhide it, but okay. What is the best model for predicting the population of Brighton? Okay, so most of you said a logistic model with limitations. A few of you kind of distributed amongst the other models. So let's take a look at the data. Okay, linear model, we can model that. We've got an R squared of 0.96, actually not bad at all, but it doesn't seem to represent the data very well. We can use a power function. It's not as good even as our linear model. Actually, but also doesn't really model the data. We could try an exponential function. That doesn't model the data very well either. We could try a polynomial fit. The problem is, actually, we get a really good fit. The problem is, got numerous parameters. We've got 3 different parameters in that polynomial equation, second order polynomial, and those parameters have absolutely no physical meaning. So it's purely curve fitting. And we can use a logistic model where we determine K and R, and actually you see that the carrying capacity appears to be about 8,000. Probably limited space for additional growth. So the model, this logistic model, is the model that makes the most sense for Brighton from a physical perspective. So hopefully this has given you a sense of the different types of models that are used to describe animal population dynamics. And I've listed the attributes and problems, and we've talked about carrying capacity.